Hello, my name is Aurélie Cobat and I am assistant professor at INSERM in the Laboratory of Human Genetics of Infectious Diseases, led by the professors Jean-Laurent Casanova and Laurent Abel at the Imagine Institute in Paris, France. I am delighted to present a project I co-lead with Professor Casanova, recently funded by the SCOR Foundation for Science. The project aims to decipher the causes and the consequences in humans of auto-antibodies neutralizing type 1 interferons, which we recently identified as strong and common determinants of severe viral infection. For almost any microbe, there is a huge inter-individual variability of clinical outcome following infection, ranging from asymptomatic cases to lethal disease. A significant breakthrough in unraveling this so-called infection enigma was the discovery of mutations in genes governing immunity to infection in patients, especially children, suffering from severe infectious diseases. In particular, defects of genes coding for proteins involved in the production of or response to type 1 interferons were identified in patients suffering from severe viral illnesses such as herpes simplex encephalitis, critical influenza, or more recently, critical COVID-19 pneumonia. Those interferons are proteins produced by our own organism to limit the replication and the spread of the viruses. This prompted us at the onset of the COVID-19 pandemics to search for auto-antibodies neutralizing those type 1 interferons. We discovered that they explained 15 to 20% of critical COVID-19 pneumonia cases, particularly in middle-aged and elderly adults. Subsequently, we and others demonstrated that these autoantibodies underlie an unprecedented proportion of severe viral diseases, ranging from 5% of severe influenza pneumonia cases to up to 40% of West Nile virus encephalitis cases. West Nile virus is a pretty common virus transmitted by mosquitoes and present in all continents, including Europe and North America. Importantly, these autoantibodies are common in the general population, with their prevalence increasing with age and reaching up to 7% in individuals over 80 years old. We can estimate that more than 100 million individuals worldwide carry these autoantibodies, putting them at high risk of severe viral infection. It is thus of major importance to determine their full range of consequences. In this context, the goal of our project is to determine the consequences of autoantibodies neutralizing type 1 interferons on severe viral infections and to uncover their causes. So we will recruit cohorts of individuals carrying or at high risk of carrying these autoantibodies, in particular patients with severe viral infections of the brain and lungs at the international level. We will evaluate the risk of severe viral disease in these individuals according to the virus, the affected organ, and characteristics of the individuals such as age and sex. Finally, another objective of this project is to determine the causes of these autoantibodies. Why do some individuals develop antibodies that target their own immune system? To answer this question, we will search for genetic variants underlying these autoantibodies by whole genome sequencing and specific analysis methods developed in the laboratory. Our program will provide novel and unique insights into the pathogenesis of severe viral diseases. It will also have major medical and public health implications. We are developing large-scale tests to search for these autoantibodies in the general population, in particular over 65 years, to identify persons at high risk of developing severe viral diseases. These persons could benefit from specific preventive measures, such as vaccination, and from targeted treatments, for example, aimed at neutralizing those autoantibodies. By clarifying the origins and effects of auto-antibodies neutralizing type 1 interferons, this project will pave the way for precision medicine.